And I am lucky enough to get to spend a little bit of time in Kona, and I will actually be officiating a wedding underwater in a little over a week. No, under a week. When is it? Thursday?
Did you forget?
Okay, so I just started a research program on Oahu looking at trying to understand pregnancy. So we don't have a huge ton of data, but I want to tell you a little bit about what we're doing. So I'm going to spend the first part of the talk just doing some background on the biology of tiger sharks. Most of the knowledge that we have of most animals is from going out and killing them. And so there was a series of culling programs in the 60s and 70s in Hawaii, and they killed a fairly large number of sharks, and fortunately they sampled them. So they kept lots of organs and everything and measured lots of things. So they generated a really fantastic database in this culling program. So you may not be very happy about the fact that they killed all these animals, but be happy about the fact that they didn't just kill them, they measured them, right? So lots and lots of really important biological information came out of what probably today we would consider to be a very unfortunate episode in the history of our state. Some of the information, this is from a paper by Brad Weatherby, Chris Lowe, and Jerry Crow. These guys are both graduates of, got their PhDs at UH Manoa, and I think Jerry Crow was Waikiki Aquarium, is that right?
Yeah, he's at Waikiki.
Yeah, okay, thank you. So they were able to go through and categorize the diet of tiger sharks and a bunch of other species of sharks that were captured in this program. They could find out things about how the diet changed as the animal grew. So the diet of little tiger sharks is quite different from the diet of larger tiger sharks. The only thing that I'm going to say about attacks on human beings today is this slide from the same paper, from the Weatherby et al. Paper. And during the period of time when they were doing this culling program, there wasn't a notable change in the trajectory of these lines of the number of attacks that were happening, which you can interpret that any way that you would like. I'm going to interpret it as an indication that it probably didn't help to kill all of these animals, because if it had helped, we would imagine that the line would have turned, right? Or at least there would have been some inflection point in the line. Anyway, some of the most important information we want to know about any species is how fast they grow, right? And at what age and size they reach sexual maturity. So it's probable that the average age of maturity, on this graph they're doing minimum age of maturity, and then on the text over there we're doing average age of maturity. So it's probably in the range of 7-ish years, maybe a little bit later. So you can kind of compare that with a lot of different species. What's the age at maturity for a bacteria? Anybody? We're measuring that in minutes, right? Age at maturity for, I don't know, what's the age of maturity for a Galapagos tortoise? I don't actually know, but pretty late probably.
50 to 75?
50 to 75 years. Age of maturity for like a green sea turtle is quite late, right? Is it like, 25, 30, something like that? So this is a relatively young age of maturity for a large-bodied animal. Age of maturity, I mean, some of the things that we utilize a lot in agriculture, like chickens and pigs, they have a pretty early age of maturity. Right? Tipsy, how are you doing? Hi. So this is, for a large animal like this is pretty good news from a management perspective, because it means that probably this species will be able to reproduce itself relatively quickly. Not fast, but it's not the Galapagos tortoise of the world, right?
What's the size or the weight of a 7-year-old?
For a, I'm I'm gonna have to refer to another, I don't wanna give you bad information. Let me, I have the perfect graph for this. Where's my doohickey?
Down.
Okay, let me try and get this thing over here. Why is my,
You have it right up there, isn't it? That's a link.
okay, let me see if I can find this. I'm gonna put this thing.
300 centimeters?
Yes.
Well, so, yeah, I'm trying to get the length weight. Here it is. I have a little cheat sheet right here. To help me remember things. Is that opening? There we go. Okay. Okay, can you see that? So our length was around 3 meters. So at around 3 meters, the weight is going to be around 400 kilograms. So what is that? A little over 1,000 pounds or something like that. So kind of like a grander marlin, right? That you might see hanging up at the dock. There's been a big marlin every day that I've been here, hanging from that scale. Wow. Okay, so I just went diving at the harbor. Just before coming over here, we saw 2 tiger sharks. They were both probably in the 10, 11-ish, maybe 12 for the second one. So maybe a little bit heavier than that. Okay.
1,200 pounds or 1,200?
Pounds, yeah. Yeah. But I mean, it's very, if you look at this graph, it's very nonlinear, right? So they don't actually have on this graph, all of the animals that we studied, most of the animals we studied were between 4 and 4.3 meters total length, which doesn't even occur on this graph. So they would be up somewhere in this region. So maybe, yeah, I don't know, like, probably like, yeah, I mean, they'd probably be in the 3,000 pound range, 2 to 3,000 pounds for those animals. Yeah. Okay.
How are they that heavy?
So, can you make your question a bit more specific?
Yes, sorry. No bone, what is giving them all of that weight? Is it muscle mass?
Well, this is mass out of the water, right? So their mass in the water is probably very slight. They're probably just slightly negative. So remember, mass is directly proportional to volume, and volume is the cube factor of length, right? That's why this graph is not a straight line, right? Because this is the cube of the length, right? So as you get bigger and bigger animals, adding a tiny little bit of their length adds a huge amount of mass, yeah, to that animal, yeah. Okay.
Yeah, but can I just add a
Yeah.
little bit? You know, there's fat girls and skinny girls.
Yeah.
So they can, the range can be very, very wide.
Yeah.
Males are typically smaller than females.
Yeah. Yeah.
But there's a huge range to them.
And so at a given fork length, like here, you can see there's quite a bit of scatter around this line. So there's a fat one and there's a skinny one at this exactly the same length. There was a difference in mass of more than 100 kilograms. So this one was almost twice as heavy at the same length. Yeah, so there can be a massive variation depending on that. Yeah. And we actually, there was one animal that we caught that we were convinced was pregnant because it was very rotund and we couldn't see any babies inside of it with the ultrasound machine. And there's 2 possible explanations for that. One is that it didn't have any babies in it. What's the other one?
Yes.
Kevin doesn't know how to use his ultrasound machine. Which is always possible, but in another fat one that we caught, we did see babies. We got pretty decent images that I'll show you later. So one possible thing is that in between pregnancies, they're going to take a year or 2 off and try to build up some energy during that time. So it may have been just that animal had been really successful at building up energy stores within its body. So it was fat, it wasn't actually pregnant. Okay. This is an image from a a fish market in Taiwan. So you can see these are pretty late term pups. They're born at about 90 centimeters length, a little under a yard. There's actually a group in Taiwan that has tried to save these little tiger sharks by putting them in an aquarium when they come out of a pregnant female like that. So because we don't have any near shore fisheries in Hawaii that are routinely capturing sharks like this, but we could get a lot of specimens if we went out in the longline fishery, although the Hawaii longline fishery now releases all of its sharks alive. But other longline fisheries in other parts of the world, you could get a lot of specimens for science by going out on those boats and sampling animals that are being taken in those fisheries. Taiwan is an amazing place to go to look at sharks. They bring in so many sharks into the market, and it is just, it is an ichthyologist's dream walking around those fish markets. It's just unbelievable the stuff that you see in there. You might be sad about something like this, but just from the perspective of seeing new species, you want new species to do fish prints on. Have you been to Taiwan? You have got to go to Taiwan. And I've got the man for you to call when you get there. Seriously, if you're going to go, I have a colleague in Taiwan who's going to take you around to all the fish markets. You probably know him. Okay, do you know Riar? Wei Chuan Xiang? Riar? You know that guy? Okay, he's a hilarious dude. Okay, so this is the paper that I just showed some graphs from. So this really is a seminal paper on tiger shark biology, and it all happened because of those culling programs, right? So very unfortunate, but lots and lots of biology came out of this. If we look at some of the things that Nick Whitney and Jerry Crow were able to figure out. So you can tell, how do you know when mating season is for sharks? Roses? Yeah.
When the willy blooms.
Lots of dudes running around flowers, okay. Anything else? Observable to divers and fishermen? Maybe the males are showing up. What's happening with the females? The females are all slashed up because during mating, the males, not having hands, the males hang on with their teeth, and the females just get all slashed up. So so very, very tough skin and very impressive healing ability in these animals because when they mate, they get really cut up. And in some species, actually, the females have much thicker skin than the males as an adaptation to that. So they were able to figure out that the mating season was sometime around in December because that's when the females were all slashed from the mating cuts. Then the thing that was a little bit bizarre is that you would imagine if you mated in December, when should your embryo start growing? Yeah, right? So you mated and you fertilized the eggs and they started growing. So, but then they figured out that the pups started growing in June or in May. Yeah, so what's going on there?
They don't have a diet to support the pups at that point.
So it's hard. I don't have a good explanation of why, but the females are storing the sperm in a special little organ inside of their body for several months, and then they're actually fertilizing their eggs in May or June, so in the early summer. And then, and so they worked this out because they had all of these uteruses sampled out of these sharks that had the lengths of the embryos in them. And there was nothing between mating season and May. But then they found these organs that store sperm in them. So they were able to figure out this whole trajectory of growth. But they also knew from observations that fishermen would have made, okay, the time of the year when I see a 90 centimeter baby tiger shark is in the fall. So we already know that the pupping happens in the fall. Well, if they had worked out that fertilization happens in June, you might imagine that they get fertilized here, they grow up, and then they get pupped here. But based upon what we know about how fast it how long it takes to grow sharks in the womb, it doesn't happen that quickly. So they worked out that it actually happens the next fall. So it's a full year plus that series of months. So it's actually about 15 months. So this is a hypothesis of, but remember the mom is in the ocean. She's weightless. She doesn't have to hold up the
But she's
mass.
gotta
of
be
gravity.
pissed off and tired and
Hungry?
you
So-
know,
Some pickles?
right.
So gestation is about 12, 15
It's
months.
15 is their hypothesis is gestation is 15. Yeah. And not only does that not line up with an annual cycle, so you could not reproduce every year, but then it appears that they take years off in between those 15-month gestation periods. So it's actually quite a few years in between litters for one individual animal.
That's a stupid question. Is it viviparous or ovoviviparous birth?
So these animals, there is not a placenta,
Okay.
and so there was a recent paper by a scientist named Jose Castro in which he hypothesized that the uterine fluid is very high in nutrients, and that was actually providing the nutrients to the pups as they grow. There's some species where the mother continues to produce unfertilized eggs, and the pups eat the unfertilized eggs. Right, so it's a very high nutrition diet. In this species, at least according to Dr. Castro, it's actually the uterine fluid that has that
Okay.
So
nutrient.
with the December mating period, I'm curious, kayak fishing. I've had years where I'll be going out sometimes 2-3 times a week, and literally every day in October I'll have a tiger shark with me.
Okay, it'll follow you around.
Where like out
Wow.
in front of Keho, or I mean, would there be any relationship there to?
Aggressive or hungry, like
No,
just
not
looking for
necessarily.
treats.
I've only been hit by one,
Okay,
and that was in like
more
January.
than I've been fine. Wow.
But coming close, yeah.
Wow.
Cruising by and hanging around and whatnot. But on the surface, right?
Okay.
So
Yeah. Wow. Did you notice if they were male or female?
they weren't rolling over for
Yeah.
me.
Yeah. I'll just go swimming next to this.
Yeah.
I have had a number of times. I have had some females, I presume, because they had their fins were really
Chopped up. Yeah.
chopped up.
Okay. Yeah.
What's the average litter size?
The litter size can be quite large. It can be 30 or more, which is very, very fecund for a shark. Most sharks have far fewer than that in a litter. Although whale sharks, did you guys see the whale shark?
Yeah.
Man, it was like 200 or something. Amazing. Yeah. That was another, I think that was another Taiwan fish market. I think a whale shark put 200 babies
So I went to Tiger Beach
in the Bahamas.
and I was there in the fall time, and it was all females, not a male to be seen. And I was wondering if they had pups maybe nearby in the nursery, maybe if that
So-
was why there's so many females, or
It's pretty common for sharks to separate there's spatially between sexes. And so who dives the harbor a lot here? How many males have you ever seen? And how many females have you seen? Can't count that high. And in our research program in December, we sampled 9 tiger sharks, and all of them were female. Carl Meyer has done a bunch of satellite tracking. He's a scientist at Manoa, and he has shown through his work that the males tend to be a little bit deeper water. And I don't know if we were to go, well, see on your blackwater dives then, right? But I'm not. So the fact that we always see females, I don't know. I think that there's possibly some sampling bias there because the harbor dive is really shallow. I'm fishing in 30 feet of water
right?
right outside of Kehi to do this research program on Oahu. So if you maybe went out into a little bit deeper water, you might find the males, because
The only time I've seen two males in the harbor were in 40 feet, and
Okay.
they were kind of creeping by.
do you remember what month that was?
That's what happens.
Because it should be in December or January, but
Usually we see
theoretically,
them in the summer,
summertime.
but
Hmm.
3,
Wow.
but only 2.
Yeah.
Ever.
The birth ratio is pretty equal of pups at birth. So where did all the males go? Yeah, I don't know. Okay, so one of the things that we would like to do in our program is figure out if this is the whole story. So theoretically we should see this size in that month with the ultrasound machine, right? And then in the future, we need to write some proposals. We would like to try to figure out where the pregnant ones are going. So Carl Meyer and his colleagues have done a ton of tracking of this species in Hawaii, but they haven't ascertained the pregnancy status before they put the tag on. So that would be the next thing that we're interested in. Okay, so can we figure out where they're going? First of all, can we just figure out if sort of this is really what's going on of what they deduced from the culling program in the 60s and 70s? Okay, so instead of going out and chopping them all up and measuring all of the babies with a measuring tape, which would be a great way of getting data, instead, we are going to use an ultrasound machine and then release them with just a mild hangover to be mama sharks into the future. Okay, so there's a wide variety of reaction to capture stress in different shark species. There are some shark species that are quite delicate. Like when Monterey was trying to collect thresher sharks. I don't know if you ever helped them out with that, but man, the thresher shark, you look at it sideways, that thing dies. And the Galapagos sharks are very fragile on the line. So
Limnatus are pussies.
yeah,
Limnatus are tough to keep alive.
yeah. So there's just a real variety. The tiger sharks actually do pretty well with capture stress. Okay, so one of the, not so much actually with this species in Hawaii because we don't have longlining close to the islands, but for species that are interacting with commercial fisheries a lot, fisheries are probably the greatest threat to sharks. We actually caught one animal that had, who knows if this is related to fishing, it could actually have just been a fight or when it was young. Where's this thing? Oh, I can just click on the picture. There we go. So you can see this animal's lower jaw is all busted. Who knows why that is? You know, maybe when it was a baby, some other shark tried to eat it and it got injured. Maybe it was fighting with another animal. Or maybe it, probably got caught in a long line and in the process of being released, you got a jaw injury. But this animal's actually doing perfectly fine. It's swimming around, biting things and eating stuff, right? It was not skinny. So they're actually pretty amazing. It can have what looks like a pretty serious injury to a very, functionally important part of its body, its mouth, and it's doing just perfectly fine. Just saw that. Okay, so a quick intro to our research team. This is our core team. We also had some other volunteers who came out. So that's my student Dan from the Virginia Institute of Marine Science. This is Brooke Anderson, and this is Hannah Verkamp, and they're both from the lab of James Sulikowski at Arizona State University. And you went to Tiger Beach in the Bahamas. So Hannah actually had to leave the project early to go to Tiger Beach in the Bahamas and do exactly the same type of work on tiger sharks over the Bahamas. So they're looking at pregnancy in the population over there using exactly the same approach with an ultrasound machine. Okay, so we're gonna go out, we're gonna catch some tiger sharks. We've been just fishing one or maybe two hooks at a time because we don't want to catch a lot of animals at the same time because that is just a disaster. With our program, we're using a pretty large, much larger than in commercial fisheries hook because we want the hook to make it all the way around the jaw and come out the cheek. if you catch these animals on a small hook, the hook goes through the jaw, and it's impossible to get it out. And those hooks are expensive. I get them all back. So for reducing injury to the animal, it sounds counterintuitive. For reducing injury to the animal, you actually want a bigger hook because it goes around the jaw and comes out the cheek, and it's a really minor injury to just have a hole in your cheek. Whereas if you poke something through the jaw, that's an injury. Okay, so we're using this giant circle hook. We're using this huge, massive leader, super heavy gear, quite different than what a commercial fishing vessel would be using, because we want to bring these animals in quicker. We don't want to lose any of our gear. And presentation is really not important in this fishery. This is not marlin fishing or trout fishing. So we can have this horrible equipment in the water and they still bite the hooks. You're never going to catch an ahi with that kind of gear, right? Okay, so we're gonna let the animal tow a bunch of floats around for probably 45 minutes in order to tire it out. And then we're gonna go and so it's just gonna be free with about 60 feet of line and a bunch of floats and a flagpole. And then we're gonna go and grab that line when we think it's tired, and we're gonna bring it into the side of the boat. And then we're going to get it secured at the side of the boat.
She's beautiful.
Oh man, these animals are beautiful. I don't know what happened to the apparently you can correct exposures on videos. I don't know how to do it, but
Sounds like a Jeff question.
yeah. So we're gonna get it at the side of the boat. We're gonna put a tail rope on it. We're actually gonna put lines on both of the pectoral fins so that we can release pressure on the hook itself, and then and we're going to flip it upside down, and so that's a pretty rotund animal, isn't it?
Yeah, very.
She's very huggable,
So let's see.
kissable.
I don't know if there might be some distortion from the camera, but it's definitely a fairly round animal. And so we're going to put a little conventional, just a plastic number tag in the back so that if it's recaptured again later on, that number can be traced to the length measurement that we took on this day. Someone catches it in 5 years' time, it's grown bigger, or maybe it hasn't grown bigger actually. It's still 14 feet long because now it's putting all of its energy into reproduction instead of growing its body bigger. But all of that information on the growth of fishes, a lot of it is coming from these kinds of little conventional number tags. This is a view of the animal irrigating its gills. So there's this idea that sharks have to swim to breathe. Well, that is the case for the more pelagic species, but most of a lot of the other species of sharks can move water through their gills with their mouths, like fishes can. So when you go and you look in fukas, you always see a white tip reef shark in there, just laying there asleep. Obviously, that thing does not need to swim in order to irrigate its gills. Tiger sharks are very good at pumping water with their mouths, and they can do that as we have them along the side of the boat. We'll also, every now and again, we'll just bump the boat really, really slow forward because that just pushes a bunch of water into the animal's mouth and keeps it well irrigated. So then, once we have the animal secured to the side of the boat, we try to find a place with calmer water. So I don't know if any of you guys are familiar with Oahu, but we're right off of Katy Lagoon here. Here's the big cranes from the shipping port. And Tripler Hospital is probably up on the hill somewhere. Okay, so this is Brooke, and that's Hannah. And you can see in her hand, that's the probe of the ultrasound machine. And Brooke is looking at the ultrasound machine. And it's so bright out there that we have to throw a blanket over our heads in order to see the screen. And so so Hannah is, so she's way up there at the very front of the animal. So she's actually trying to get an ultrasound of the heart. So this is part of the project that we just started now, and actually I'm hoping that they did this when they went over to the Bahamas, because nobody's ever measured heart rate in these animals before, and so we just like, do you know what the heart rate of tartar is? Anybody? No, we don't know, and we can do it non-lethally, right, without harming them with an ultrasound machine. So while we're trying to get this pregnancy information, we can also get some heart rate information. So underwater, this is a view of Hannah putting the ultrasound probe onto the animal. And
That's where the heart is, like that far.
it's way up there, yeah. So it's right where the cartilage of the that is supporting the gill arches, where those two bits come together right here. If you trace those up and stick the probe right where those things come together right here, that's where the heart is, and you can angle it a little bit that way. Those are all going to be pros at measuring heart rate.
Send us, friend.
And then every biologist who ever touches any animal cuts a little tiny clip of skin off of it. He keeps it as a DNA sample, right? So we do that. So cut a tiny little clip of the skin of the fin off, stick it in a tiny little plastic bottle of preservative, and send it to one of our genetics wizard colleagues, to do it. Of course, anyway, so that's kind of just a normal thing. If you're gonna cause so much inconvenience to this animal, try to get some useful samples. Okay, and then. So this is taking the hook out. Actually, so did you see in the previous one, did you see in the previous one the way the hook was going through the corner? And you could actually see it didn't go through the jaw, it went through the cheek. So with my brand new welder, Shane, we created this tool where we can grab the hook, and this is a circle hook, and we can use the leader to rotate it around and then push the hook out of the shark's mouth. And so the hook just came out right there. And so we don't have to do something really awful like cutting it out. Like actually, if you cut the cheek and pull it all the way through, that's a really minor injury. But if you just like hacksaw through the jaw to get your hook back, that's a terrible injury for the animal. Okay, so.
Is a shark on just because it's upside down?
Oh, so I forgot to. Thank you very much for that question. I forgot to. So who's ever flipped a shark upside down? Yeah, and did it work? Did it calm down?
It does.
Sometimes it
Sorta.
doesn't. So it worked really well in this species, at least at this size class. It probably wouldn't work as well for the little ones, but so there's this phenomenon in a lot of species that scientists have termed tonic immobility. When an animal is turned upside down, it will calm down. It happens in various mammals and some birds. It happens. Sorry.
Flounder.
Flounder? It works in flounder, really?
Halibut.
Wow, I did not know that. So there's even papers about tonic immobility in human beings in the scientific literature.
What?
I don't know very much about that, but when I was searching that search term, and a bunch of papers about human beings came up. Okay, so given now what we know about tonic immobility, calming the animal down, just skip. Whoa. Skip forward just to show you one thing. Oh, no, wait, I need to go over to here. Oh, I can't even read that. Okay, so this is some ultrasound.
This is so cool.
Okay, this is an ultrasound of, can anybody guess? Can I make this bigger? See that? Okay, that is an ultrasound of the heart.
Yeah.
That's the ventricle of a tiger shark. This is 21 centimeters deep into the animal, right? So it started at about 4 centimeters. This is a big heart. It's really big, right? This is the scale in centimeters. Okay, this measurement was taken when the animal was upside down in tonic immobility.
Right.
Okay, what do you guys think about this data?
Pretty awesome.
It's brand new. But
It's a starting point. It is
It's gonna
just the
speed
starting
up
point.
as soon as
This
you
is
flip-
probably the low.
Yeah.
So I haven't figured out a way yet to get this for these large animals. As soon as we flip them right side up, they're just like, it's rodeo time.
Right.
So this, we've done this on little tiny baby sandbar sharks in Virginia. And at the time we were doing it, I didn't do it with them right side up. And then as soon as we started telling people about our results, they were like, what is wrong with you? So that is a piece of information that we still need to gather. It's not going to happen on these big tiger sharks unless you guys give me a good idea about how we can get that done.
Give me a big tuna.
Okay.
So is it like 25, 30 beats per minute?
So slow. Yeah. Isn't that incredible? So slow. That's like slower than, who was that star tennis player who had the crazy low heart rate years and years ago? Can't remember.
Wait, so what was the initial heart rate? That you
It's
found in the...
we're getting between 20 and 50. per
Oh
minute.
wow.
Super slow. Yeah. But again, this is the animal upside down in tonic immobility, so who knows what's happening.
Right.
Okay, so this is just an image of us trying to irrigate this animal's gills. So we're driving the boat forward, and we're trying to make a whole bunch of water go through the animal's gills. And so I don't know if you can see it, but you can kind of see the gills flaring out.
Yeah.
See how much they're flared right there?
Yeah.
So there's a lot of water going through. So we're trying to keep some oxygen in this animal's body as we're giving it a hangover. And then at the time that we're going to try to release it, we're going to hold it. We're going to take the hook out and take the ropes off and everything, and then just try to get it up to speed a little bit by holding it there and bumping the boat slowly forward, so that when it starts going, it's already got some speed, because they're very disoriented after they've been abducted by aliens. The worst possible thing you can do is have an upside down shark and just let go of it, because it'll just go. It'll just sort of sink. And I've never jumped in and swam after it, but do not do that. Who knows if they would at some point, maybe they would wake up, but it does not look good. Okay, so here is the figure from Nick Whitney and Jerry Crow's paper. So we sampled in December, and so if we can find a pregnant female, how big should the pups be in December?
It's like 23 centimeters.
Yeah, 20-ish, 20, 30, something like that. Right? Does that make sense? So this is the length of the pup, and this is the month of the year that the uterus was sampled from in the culling program. So if we sample in December, if we follow this up and across, that's about the size that we're expecting. Right? Okay, so this is what we found. And again, remember, this is the scale in centimeters here. So how big is that tadpole? How big do you guys think that tadpole is?
Where's zero on that?
Where's zero on
The top?
The 0 mark is this very.
Yeah, absolutely.
This is the 0 mark. This is awesome.
Hmm.
I thought it was going to be a chopstick.
No, that's our pointer. Is that a,
It's the pointer.
yeah, that's what we use.
Is it like
12?
Yeah. So 5, 10, 15, 20, 25 centimeters.
So 15.
How many centimeters in a foot?
30.
30. So a foot would be down to here, right?
Okay.
A foot long would be down to there.
I know.
So from there to the bottom of the TV. So it's about right. Yeah? Is that reasonable to you guys? It's not, so, because like,
Point down to the end of that.
okay, oh, sorry, okay, so this is the snout. That's an eye. That's an eye. These are gill arches right there. This is the spine coming back into the tail. And according to me, this is part of the tail of the same animal.
Oh.
Yeah.
See, I thought it ended
So
right.
this is a still. Let me show you guys some moving ultrasounds. So I believe that the end of this animal is possibly somewhere over here, which would put it at about 20,
Okay.
maybe even longer than 20 centimeters. Okay. So here's another still. So this is the lower jaw, that's the upper jaw, and that's the snout.
Oh, that's so cool.
Yeah, and so you can see that this is the body coming down here. Okay, so if we can figure out what the pregnant females are doing, it would be, at least to me, it would be interesting to know if they're doing something different than everybody else in the population at the time that they're pregnant, particularly in that run up to when they're going to have pups in the fall. So it'd be really, really interesting for us to be able to, once we have tried to figure out a pregnant one, to actually try to put a tag, like a satellite tag on it. Okay, so Carl Meyer and colleagues at UH Manoa have done a ton of work on this species over a 20-year period. This is one of their recent papers showing the home ranges of individual tiger sharks. So in their kind of normal everyday life, their range is about the scale of like one side of Oahu. So that's like, 20 kilometers, 30 kilometers, something like that, right? That's what these shaded patches are. The red is where they're really spending the most time, and then as you go out to the blue and the gray, or the lavender, is that lavender?
Yeah.
Wouldn't that be a great color for a shirt, that lavender color? So this is kind of their normal everyday commute, and then Carl and company have also shown them swimming between the neighbor islands up to the Northwest Hawaiian Islands, from the Northwest Hawaiian Islands to the main islands, thousands of miles out into the middle of the Pacific Ocean. They've shown all of these movements. So it would be really, really cool to continue this type of work, but to know that one was carrying babies, and that one was not. So that's what we would like to do in a future version of this. Okay, so if I can, yeah.
Okay, all of my research experience has been with mammals, so
Okay.
please forgive me. I'm trying to compare.
You need
animals
way
and
more
orchids.
equipment.
Yeah, a lot. We will go there. But my question is, do we see like a insular population and offshore population, or is it every shark everywhere? Or do we not know? Like,
So Carl is the person to ask, not me, of that question. But from reading his papers, there are kind of animals that spend most of their time in the Northwest Hawaiian Islands, but they'll swim to the mains in order to breed,
okay.
and then they'll go back. And then there's animals that mostly hang out in the main Hawaiian Islands, and that area of Maui, where unfortunate things have happened, there's just a lot of animals that live there. That's what Carl and company hypothesized about that is that the whole Maui Nui platform, including Molokai and Penguin Bank, is very unusual habitat for the Hawaiian Islands, right? Normally in Hawaii, especially for you guys here in Kona, right, you go a couple of 100 yards off of shore and it's like 1,000 feet deep, right? But over there in Maui Nui, you have pretty shallow habitat that's very, very rich habitat, tons of bottom fish, just lots and lots of fish biomass over there, so probably lots to eat for a tiger shark. That's their hypothesis of why it seems like there's more tiger sharks living there than in other parts of the Hawaiian Islands. Okay, so let's see. Okay, so here is here's the little, so this is like a plan view, like either a view from either above or below. I'm not really sure which, but we're getting this view.
How many would you say are in that?
So we didn't keep the animal long enough to do a really exhaustive count, because there could have been like 30 of them in there, right, or something like that. There were definitely embryos in both uteruses, and the ones that I saw, they were all about the same size, which is not surprising, right? They all probably were fertilized at the same time.
How do you, when you're scanning, do you have a grid pattern you follow or something?
So for this we start at about the point of the rear tip of the pectoral fin, and then just slowly move backwards. So it's on a large animal. It's it's a distance about that long. It's probably uterus is probably.
But you're only covering this much. Are you going left to right and then like that?
It's like kind of like start about here and then just kind of go down like this, and occasionally like go back and forth looking for stuff. Part of the challenge is that there's one person looking at the screen and another person holding the probe. There's fancier ultrasound machines that come with a set of kind of goggly things. And I don't have that ultrasound machine, but that makes it easier because the person holding the probe can see what's going on.
And do you snapshot the grid, like to review the sonograms later?
We just record a video as we're going.
Okay.
Yeah.
So the display is real time. I mean, that's the time. Is that your scan time that we just watched, or is it accelerating and decelerating?
That's the real time. That's in real speed. And so, so.
And then we're looking at the length of the uterus. We're looking at the scan as it moves.
So these videos were taken while the probe was not moving, because basically we're like moving the probe. So when this happened, I was looking at the screen, and my volunteer, who's 7 feet tall, who has a huge wingspan, I put him on probe duty because you have to reach. Instead of having someone lean way over the side of the boat, he was able to just reach with his eagle wingspan. And so I would just yell at him, stop, right there, move a little this way, move a little that way. And then when we got what we thought was a really good image, we would hit the button and it would record a 60-second video. And then we would move again. So I actually don't have a video of what you would see if you did the moving scan. I should, and I don't. I'm sorry.
So you've got a bunch of samples, basically.
Yeah, it's like when I thought I got a good image of an embryo, I would stop and tell him to hit record. Okay, so this one. So can you see this one up here on the left?
Yeah.
So it's
Teeth, like the sand tigers as an embryo?
they do have teeth. Yeah. You might almost be able to see them in it. You can kind of almost see teeth. Yeah.
Yeah.
So this animal is pumping amnion through its gills to oxygenate its body, and if you read Jose Castro's paper, it's probably actually swallowing a little bit of that fluid in order to gain nutrition, according to Jose Castro. There's some other stuff going on down in here that might have something, it's very hard to, I need to go to medical school and take an ultrasound class.
So have you shown those to a radiologist?
No. Is there a radiologist in the room?
Oh, no.
I will say.
You have one coming next week
then.
So I do have a colleague who is a brain surgeon who uses ultrasound a lot, and I have been meaning to go to his house with the ultrasound machine and get some lessons.
I'll ask the radiologist if anybody is interested, I'll send them
Okay.
away.
Oh, that would be fantastic.
Cause we have few here on the island.
Really?
Loves to do this kind of stuff up in Waimea.
Really? Ooh, wow. Trip to Waimea.
Yes.
So there's a couple visible in that image. There's more than one visible there. So you can see on the right side, you can see that other one is pumping its mouth. And there's probably, this is probably another mouth right there. So I think there's 3 at least in that image.
Do any of them eat each other?
Not in this species. In the
sand
sand
tiger
tiger
they do.
shark they do.
Yeah.
And then only one gets born.
Right,
Um,
right.
Or one from
It,
each uterus.
it really
It
is.
can be 2, one from each uterus, 2 uteruses.
Oh,
Yeah, but only one.
That's
in the-
true survival.
yeah.
Yeah. So this must be a view from the head, and the other ones must have been a view from the belly, because now we're seeing like the gill arches and the eyes and everything a little better. When there's any motion, it's just impossible. When the water is rough, it's really, really hard. So we try to find calm. But yeah, so you can see those gill arches and everything.
Whoa.
Yeah. Yeah.
No,
So-
we're not nerds in this room.
So, okay, we're pretty much out of time. Let me just show you. So the reason that heart rate is interesting. So these were our heart rate ones. Whoops, it's no longer big. Well, you guys saw the heart rate one, right? How do I get my PowerPoint back? Oh, it's on the PowerPoint. Okay, so if we're really interested in heart rate because it can tell you a lot of stuff about the biology of the animal, and it's particularly if you could get a time series of heart rate. You know how our smartphones now are just snooping every aspect of our daily lives? So the accelerometer in your smartphone is telling Big Brother a hell of a lot about you, right? Because every time you touch that thing, the accelerometer is measuring the shaking or the motion. When you're running, it can tell you're running. When you're walking, when you're sleeping, and you're just rolling gently, it can tell that, if you had your phone on you. So they can learn so much about that. Well, your heart rate is also changing a lot with everything you do all day long, right? And in addition, when you eat something, it takes a lot of energy to digest a meal. So we don't eat before, that thing, don't eat before you go swimming. I always eat before I go diving because I know I'm getting cold, right? So I need some energy. But a lot of blood goes to your gut when you eat a meal. That takes up a lot of the oxygen supply of your body. We can only pump a certain amount of oxygen through our bodies. And if 30% of it is getting used in the tummy, that's 30% that's not available for escaping predators or catching prey, right? So this digestion of a meal, you can see in the heart rate of a shark. So, a little arrow thing. Can anybody see it? Oh, there it is. Which side of this screen is? Oh, there we go. I didn't know which the relationship of the two screens was. Okay, so I actually was able to get a time series of heart rate from a little tiny tiger shark. I mean, a little tiny sandbar shark, which is this. So we put in a heart rate tag into this shark, and the black dots are the heart rate measured by the tag. The red line is the temperature, and the black arrows are when we fed the animals that were in this tank. So these are captive animals in the tank. Okay, so this is, we captured the shark and we did this procedure. It was very upset because we had captured it. We threw it back in the tank, high heart rate, 80 or something like that. Calm down, calm down, calm down, calm, 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 calm. Ate a meal, right? So this is the heat of digestion.
That's not the feeding frenzy heart rate.
Well, so there probably was a feeding frenzy, but this is a 3-day period. So that feeding frenzy took 20 minutes when we're feeding the tank. and then its heart rate was elevated for 3 days after that while it's digesting the meal. So we could actually tell when this animal ate. So if you could get a really long-term record of this, that would be really amazing, because we don't actually know the feeding frequency of most marine animals, actually. So that's kind of the one of the ultimate aims of this work. The other thing is,
Why does the heart rate get so crazy?
Okay. The reason the heart rate gets so crazy in this part of the record is that my tag is not in the right spot anymore. So this is all garbage over here. Thank you for asking that question. So far we've really only managed relatively short periods of time of good data from this. I mean, as soon as you start getting this wild scattering at exactly the same time, you're like, well, in that 2-minute period, do you really think that the shark had a heart rate of 150 and then a heart rate of 40? No, definitely not. So the right third of this graph, the tag had slipped, and the data is total garbage. One of the other things, this is my pet hypothesis that I haven't managed to get a proposal funded to look into, but if an animal is dying, its heart rate is just going to get slower and slower and slower and slower, and then go to zero and it's dead. So for estimating how many of the sharks that interact with a commercial fishery die as a result of that interaction with the fishery, at the moment, what scientists are doing is they're putting satellite tags on sharks and letting them go. That costs about $5,000 per shark to tag.
Wow.
Right? Very expensive. You're not going to have big enough sample size, right, that you want. They're also taking blood samples, and we can get measurements from the stress, measure things like lactate in the blood to get an idea of whether the animal's going to die, right? So that's another method. Another thing we could do is we could take a measurement of their heart rate. Now, I haven't demonstrated whether this would work yet, but my idea is if we can ground truth it perhaps with the blood and the satellite tags, and we can actually show, any animal that has a heart rate below 20, toast. Then we could go out on a bunch of commercial fishing trips and measure a bunch of heart rates and get a large sample size of what percentage of the interacted animals actually ended up dying. That's really important to go into the stock assessment, which is going to be used to manage that population. So that could actually potentially have some use, rather than just being a gee whiz kind of thing. Yeah.
In the 80s, we looked at blood gas chemistry, and you can get, you can measure CO2 levels and oxygen levels, and you might want to look at that as a baseline.
I haven't measured CO2, but that would be an excellent thing to measure. I don't have the equipment to do that.
You can buy them surplus now. They're not that expensive.
Really? Okay. I should talk to you about this. We were measuring lactate, glucose, and hemoglobin, because those are very straightforward to measure. I had a machine for a while called an iStat, which is a veterinary analyzer that does a whole bunch of things. And for about, 5 or 10 years, people just iStatted the hell out of a bunch of sharks and fishes. And then somebody actually busted the machine open or maybe read the instruction manual and said, well, this thing raises the temperature of the sample to 37 degrees centigrade before it runs the analyses. Because it's designed for your dog, right? It's a veterinary, or a horse, or a person, right? It's a veterinary tool, we could just as well use it on a person, right? That completely throws off a large number of the analyses that you're trying to do. So then a huge number of papers were instantly garbage. Actually, one of my graduate students, we can't publish as part of his thesis because he used the iStat, and we didn't know at the time that he was doing this, and then all these other papers got published. Oh, okay, sorry about that. Throw that one in the garbage can. That 3 years of back-breaking labor doing those experiments. Yeah, so that's kind of the end game.
Would we expect to see the same heart rate across species?
Across different shark species? I would doubt it.
Okay.
I think it would be quite species
So
specific.
if you went out for 6 say you do this commercial fishing, you still have to do it species.
You have to do it species by species. The blood chemistry that we were talking about has to be done species by species. It's very species specific. Yeah. Yeah, and the thing about the Galapagos and the dusky sharks dying really easily, Heather Marshall has done a bunch of work on this, and they have some weird thing that happens to potassium levels in their bodies when they get stressed. And they die at very high rates, much higher than a lot of other species. So there's lots of stuff going on with the physiology of the animals that's very species specific. Cool. Thank you.
Oh, thank
Yeah.
you.